Hello, everyone, and I'm very honored to be here. I think I'm the only non-professor speaker today, actually. I'm a medical intern at this hospital and defended my thesis under Eric's supervision about 1.5 years ago. And I will talk to you about neuroglia and why they are important, especially for sleep and neurodevelopment. So one can say that neuroglia is sort of a forgotten cell type or a group of cells, and I hope there will be a change to that, especially after this talk. I'll start out with some brief history, and then I'm going to try to convince you that this is an important cell or cells. We'll talk a little bit about their role in brain development, and then go into about sleep regulation and finish off with some white matter injury, which I think all of you know more than me about. So glial cells is not a newfound thing. They were discovered by this guy, Rudolf Virchow, about 150 years ago. He found the cells and termed the coin Nervenkit, or what was then translated into like semi-Greek to neuroglia, where glia means glue, so something that holds the brain together. The idea was that neurons are the brain and the glial cells make the neurons stick together and function as an organ. He defined them as electrical non-excitable cells that were not blood vessels, so to differentiate them from the neurons that everyone was so interested in. Then a few years later, Golgi, who did a ton of neuroscience work, suggested that they might have some metabolic supportive role since they were found in very close proximity to the blood vessels. And in the early 1900s, Ramon y Cajal selected that maybe they insulate the neurons as well, because they are both in close proximity to the blood vessels and the neurons. So essentially they are everywhere in the brain. Now, 150 years later, we know that they have this structural homeostasis role. They are the glue of the brain. They link blood vessels and neurons. They assist during the neuronal migration during early brain development. They are essential for forming synapses to connect the neurons in the brain, and they produce myelin. So they also insulate the brain. So all that was discovered in the 1800s have been shown to be true. But now we also know that they are an energy reserve in the brain. They regulate the entire cerebral blood flow. So they have huge metabolic roles. They are part of the defense of the brain. The immune cells of the brain are a type of glia, microglia. So they buffer free radicals and form the connections with the rest of the immune system to recruit more immune cells when needed if you get an infection in the brain. They're involved in blood gas control, so they regulate the levels of oxygen and carbon dioxide in blood by modulating respiration. And they have some neurological functions. They are involved in circadian rhythms and controlling the respiratory rhythm. So what are the cells? Well, you can define them as macroglia and microglia, which is what we'll talk about today and focus on today. Macroglia are astrocytes and oligodendrocytes. I'll come back more in detail on those. Then there are Ng2 plus cells, so polydendrocytes, and radial glia, which are a bit obscure. The Ng2 plus cells, no one really knows what they're doing, but they're in the brain. And radial glia is only active during early brain development, so I'll skip on those today as well. What is interesting is if you look up until 2010, almost all neuroscience has focused on neurons. Very few papers have focused on the glial cells. So we have a bit of catch up to do. Another interesting perspective to look at this slide. So I don't know if you will agree with me, but I'll try to convince you. So down here we have sponges. Not much of an animal, really. They belong to the animal kingdom, but they don't have a nervous system. And if you go up in this slide, you end up with the roundworms, and then you have snails, and then some starfish. You end up with the chordates, where you have the vertebrates on top. So you could sort of say that this is, the higher up in this scale you get, the more advanced you are. especially in terms of neurodevelopment. All right? The thing is, the neurons down here function the same way as the neurons up here. What is different is the glial cells and how advanced they are. So down here, this first around the roundworms, where neuroglia actually becomes needed for neuronal survival. And up here in the vertebrates, we have deep specialization of astrocytes. There are very different types of astrocytes with different functions. So the neurons are essentially the same, but the glial different. So, is this what makes us smart? And this was investigated a few years back, where a group of researchers took human fetal glial precursor cells, so cells that are taken in early development from aborted fetuses that are destined to become glial cells, especially astrocytes. And then they took these and put them in the brain of newborn mice. All right? What happened was that in the control mouse here, where it hasn't received any human glial cells, everything is red. I know it's terrible to compare red and green, but that's how they stain the cells. So everything is red in the control mouse. After 3 months, you can see more green, and green is human astrocytes. So the longer it gets, so after 12 months, a full year, there are no mouse astrocytes left in this mouse brain. The human astrocytes completely take over and buffer out all the mouse astrocytes and take control. So there are a lot more human astrocytes compared to the mouse astrocytes. And what happens then? Well, these mice start to learn better and faster compared to their siblings that have not received human astrocytes. So in fear conditioning, you teach the mouse to associate a certain tone, a beep sound, with a tiny electrical shock in the foot. So each time it gets the electrical shock, it will go and freeze. And then when it hears the tone, it will freeze because it believes it will gonna get a shock, it's like Pavlov's dogs. And the percent of freezing increases much more rapidly in the mouse that has human astrocytes compared to the mouse astrocyte mouse. So they learn better and they learn faster, and they also can distinguish which cage they get the shock in. So if you teach them that in this cage you will get a shock when you hear the tone, they can distinguish that and not freeze when they hear the tone in a different context. So they get more advanced learning as well. And then if you look at memory testing, so you have this Barnes maze. What you do in here is underneath one of these holes, there is a platform that the mouse can rest on. And mice don't want to be exposed like this, because then a bird or something is going to come and grab them. So they want to hide. And what they do is they seek these holes and then they find the platform. After a while, they will learn and remember which hole the platform is under. If they have mouse astrocytes, they go quicker into the correct hole and find the platform, and they do less errors. So human astrocytes improve the memory of the mice. And then finally, they looked at object recognition, which is also a test for sort of cognitive functions. And what they see here is that the chimeric mouse, the mice that have the human astrocytes, thinks new objects are more interesting. So they say, okay, I've seen this object before, I don't have to bother with this. However, this one is new, let's check it out. So they tend more interested to the new things, and if you inhibit this astrocytic function, sort of, they perform the same. So the astrocytes are really important for basal cognitive functions. So I hope I have convinced you that this is an important cell type now. This is sort of what makes us smart. But what does it actually do? So during early development in the brain, the astrocytes are required for keeping the homeostatic control. They want to keep the neurons alive, and they want to have produce a suitable environment for neurodevelopment. The neurons can both proliferate and differentiate, and the astrocytes make sure that can happen. They also secrete a lot of axonal guidance molecules so that the axons can get to the correct region. So it helps guide where all the brain connections can go. And then it also regulates the vasculation of the brain. As I said earlier, in the adult, even the astrocytes regulate cerebral blood flow, and during development it regulates where these blood vessels should be. So if you have a region with high activity, the astrocytes will become very active and call upon more blood vessels to make that a more sustainable region. And then it regulates the synaptic formation and which synapses that will survive in the end. And this is an important thing because it goes on even postnatally. So most of these things, this is early brain development to establish the brain, but then the astrocytes keep making sure that the correct synapses are still there even after birth. So without the astrocytes, you will have less and sort of bad neuronal connections. And how do they do this then? So I will go through this set of 3 slides. This is how astrocytes talk to each other. So the big field here, that's like greenish yellow, that's an astrocyte. And then we have another astrocyte here and the third astrocyte over here. And you see they are close connected to the blood vessels. That was suggested already in the 1800s. And here you have a synapse, 2 neurons talking to each other. And over here you have another synapse, 2 different neurons talking about something else. And this first neuron say, hey, how are you doing? And then the astrocytes react. Okay, something is going on here. 2 neurons want to talk to each other. The astrocytes react and start to raise their calcium levels. So calcium is a way that astrocytes can talk to each other. And this calcium can then spread to the next astrocytes that's connected to it through these gap junctions that Eric mentioned, and propagate the signal through an astrocytic network so that it eventually reaches the other synapse. So the astrocytes make sure that this synapse over here, this couple, knows what these first guys are talking about. So they can modulate the signals and propagate them over large distances. They're not as fast as neurons, but they're very efficient anyway. So they can reduce or increase the activity here depending on what was said in the beginning here. And doing that by regulating neurotransmitters, the presence of ions, how much glucose the neurons will get to make sure that they function the way they should. And this part of development is really important because if you look at different syndromes like Rett syndrome, which is a sort of autism spectrum disorder, Fragile X syndrome, depression, schizophrenia, various cognitive disabilities. Many of these are associated with defective astrocytes. So the neurons in the brain are actually functioning the way they should, but the astrocytes are not. So then they get the baby in the end gets cognitive disabilities and doesn't have a brain that developed normally, although the neurons did. So the astrocytes are really important for basic brain development. Now moving on to the next type of glial cells, the microglia, which is sort of the odd one out in the brain. So microglia, they differentiate from hematopoietic stem cells in the yolk sac, a little bit from the neuroectoderm, but that is in parentheses. So they're not neural cells like all the other cells in the brain. They're immune system cells. They can differentiate into macrophages when needed and take care of all the defenses of the brain. During development, they can exist in both latent states and activated states. They can sit resting, and then when needed, they become activated and start eating things and calling upon other cells and modulating their microenvironment. What they do is that they in development, they migrate to the brain really early. So already in the 4th week of gestation, the microglia starts invading the brain, like when the brain is open, before all the neuronal proliferation has occurred. And then it reaches its peak density around week 20 to 24. So that's when we have the most microglia in the brain of our entire life, right around where the most preterm babies are born. So they have a lot of microglia, and we need to remember that for later. They have a very dynamic activation pattern, meaning that they stay latent when they're not needed. And then in regions where there's a lot of activity going on, so I mean, not the entire brain doesn't develop simultaneously. Regions develop in different orders. And when a lot of proliferation, differentiation occur, that's where you have a lot of activated microglia. What they do, they clean up after the neurons and the astrocytes. They make sure that the connections not needed are disrupted, and then eventually the cells not needed are eaten. They induce and maintain the apoptotic activity of neurons so that the correct cell, the correct neurons end up in the correct place. If you have a neuron that's not functioning in one place, the microglia make sure that that cell dies. And this way it also stabilizes the amount of neural precursor cells, that you don't get too much of a brain, because that's not good either. And then after birth, term birth, they continue to modify the synapses and get rid of the synapses not needed. And the third type of macroglia I'm going to mention is the oligodendrocytes. I guess a lot of you have heard about this. These are the ones that produce the myelin or insulate the brain, that make sure the axons function as electrical wires, that the signals can propagate really fast. Without the oligodendrocytes, this doesn't work. And this is a short video of a mouse that doesn't have functioning oligodendrocytes. And you see that the movement pattern of this mouse is not normal. It shivers, continuously having locomotive activity. It can't stay still, because all in all, it goes on electrical activity in the brain that just spills over to other neurons, because there's no insulation anywhere. So it just goes shivering all the time. So the oligodendrocytes are really important for a stable brain function. What do they do during brain development? Well, around week 17 to 20, the oligodendrocyte starts to differentiate and prepare for myelinization. Then the actual myelinization starts around week 26 to 28. So this is an interesting time frame to think about, because if you have a preterm baby born week 24, 26, the myelinization process is just starting. So they will be a little bit like the shiver mouse, all right? They don't have the insulation needed for proper neuronal function. And then this process, it goes on up until our 20s. And it's one of the things that is sort of affected by alcohol, which is why you shouldn't drink alcohol before the myelinization process is done. The process in itself is stimulated by electrical activity, so non-functioning neurons doesn't get wrapped by the myelin. So it's an efficient process. Then in adults like us, it's very limited formation of new oligodendrocytes. So the ones we get very early in brain development is the one we have to stay with for the rest of our lives. We shouldn't lose the oligodendrocytes, because we can't really get any new of them. And if you get demyelinating diseases, you can get some of the function back and proliferate a bit of new oligodendrocytes, but they don't function as well as the oligodendrocyte precursors that you have early in brain development. So it's very important to take care of your oligodendrocytes early. So to summarize the glia neurodevelopment, we have the synaptic formations. One neuron has to find the other, and it doesn't do that without glial cells. The astrocytes make sure that these two find each other, and the microglia make sure that the ones that doesn't find each other doesn't mess things up. So they clean up after the process. This is the main process that I do that. They find the neurons that hasn't a close connection. They send out eat me signals that the microglia react to, and then make sure that the cells die. And then during all brain development up until we're adults, or even after we're adults, the glial cells modify the neurotransmission by regulating neurotransmitters like glutamate, and it takes up and modulates glucose availability for the neurons. It stabilizes the metabolic state. So I hope by now I have convinced you that glial cells are important and that they have a tremendously important role during brain development. Because now we're going to go into what happens in a little bit about preterms and what's the theme of today about sleep. So what I'm not the first one to mention today, but adenosine is one of the main neurotransmitters that drives sleep homeostasis. An accumulation of adenosine makes us tired, and as has been mentioned several times as well, is that caffeine is an adenosine receptor antagonist. That's why we get so non-sleepy when we drink a lot of coffee. And what's the theory that this is because neurons release ATP when they signal, and ATP is broken down into adenosines. Like the more neuronal signaling you have, the more ATP you will get, and then you will eventually accumulate more adenosine, and then you get tired. The problem with this theory is that the neuronal signal is millisecond based, so it's way too fast to actually explain the accumulation of adenosine. Instead, astrocytic ATP that is released leads to a presynaptic inhibition in the brain. So astrocytes, as I said before, are much slower in their signaling. So it will release more ATP over a longer period of time so that more adenosine can be accumulated. So if you have a mouse where you have where you inhibit the glial release of ATP and other glial they will actually have a higher, called an FESP slope, which is sort of an accumulated measurement of electrical signaling. So they will signal a lot more if you inhibit the astrocytes. All right. So in the wild type mice where you actually have functioning astrocytes, eventually you will dampen all the neuronal activity in the brain. And what happens if you do the other way around? Instead of inhibiting the astrocytes, you stimulate them. If you do that in the cortex, you will induce slow wave oscillations, which resembles more of this is in mice, a sleeping pattern. They don't fall asleep, but they get a cortical activity that is sleep associated. And if you stimulate astrocytes in the hypothalamus, it will have like an antihistaminergic effect. You know, if you take antihistamines, like old antihistamines for hay fever, you get sleepy and drowsy. Stimulating astrocytes in the hypothalamus have a similar effect. So in these mice, you actually, when you stimulate them, you get a higher time spent in REM sleep and non-REM sleep, and a reduced time spent awake, just by modifying the astrocytes. The neurons are not touched, directly at least, because what the astrocytes do is that they modulate all over neuronal activity. So astrocytes have a key role in sleep. They also have a key role in circadian rhythms. And what was mentioned earlier is that the neonate and the premature babies, they don't really have the circadian rhythms. They have these ultradian rhythms because the glial development is not finalized yet. So this is data from adults, just to exemplify to you that the astrocytes and the glial cells are very important. So you have the nucleus suprachiasmaticus that was mentioned earlier. This is where the circadian rhythm is controlled. In this region you have cyclic gene expression and cyclic electrical activity. So it goes on and off in a regular pattern. And what you can do is you can entrain this region by light. So when we get sunlight, we sort of resets our circadian rhythm to let us know like when we're supposed to sleep and not. And this effect is partly mediated by astrocytes. So light induces an astrocyte activity. So astrocytes are directly modulated by light and thus sets the clock. And if you look at the molecular rhythms in the nucleus suprachiasmaticus, they drive the electrical rhythm in the neuronal pacemaking. So this is gene expression in astrocytes, the molecular rhythm of the region. So you see certain genes are active rhythmically by 24-hour period. So some genes are only expressed during night, and then they're silent during the whole day, and then they're expressed during the night again in astrocytes. And this is also true if you look at calcium signaling. As I said, astrocytes signal a lot by calcium signaling. So astrocytes in this region have a rhythmic calcium activity with increases during nighttime. So during night, calcium levels are high in the astrocytes, and during day they are low in astrocytes. On the contrary, neurons are active during the day, and if you mess with the astrocytic gene expression in this region, you will change the activity pattern of sleep and how the mice behave. So a summary image of this, during night, a lot of calcium in the astrocytes that will close glutamate channels or reuptake pathways. So you have a lot of glutamate in this region, which will stimulate GABAergic neurons. And GABA is an inhibiting neurotransmitter. So by not taking up glutamate, you will have an increased inhibition because the astrocytes have a lot of calcium. During the day, we have the opposite effects. They have low calcium, which opens up these glutamate reuptake channels. So they will suck out all the glutamate here, and then it will have less GABAergic tone and more activity. So the astrocyte calcium levels regulate neuronal activity during day and night. Next, about what astrocytes do during night is the glymphatic system. So the glymphatic system was mentioned earlier. It's similar to the lymphatic system, and it moves cerebrospinal fluid. So cerebrospinal fluid will circulate around the blood vessels through outside the capillaries, and then actually be uptaken by astrocytes and then transferred to the environment around the neurons. And then, so this system through the astrocytes propagates this flow. And that is important for macroscopic waste clearing. As was mentioned, beta amyloid will accumulate in the brain if this system doesn't function properly, and then you will get Alzheimer's eventually. But it also is important for distribution of glucose, lipids, amino acid, everything that the brain needs, essentially. What is interesting is that the glymphatic flow is reduced by 90% when you're awake. So it's almost entirely active during sleep. And this doesn't, when you look at the mice, it doesn't matter if they sleep naturally or if you anesthetize them. If you anesthetize them, the activity will go up. So this is a system that is active when you're sleeping, no matter why. It's not dependent on circadian rhythms at all. It's only sleep-wake states. partly is this because the flow is suppressed by norepinephrine, which is has high levels in the brain during activity and during day. And what happens if you get disturbed sleep, you will have reduced activity in the glymphatic system, and then you will accumulate neurotoxins and the brain doesn't function properly. So you have to sleep to keep this system active, and if this system is not active, your brain doesn't function. Simple as that. All right, final few slides. White matter injury. So white matter is essentially glia, so it's quite of a kind of an open goal to talk about white matter injury when you talk about glial cells. What is interesting is that the areas with the most microglia during brain development are also the regions that are particularly vulnerable to injury. So the regions that you have a lot of active microglia will accumulate, or that's where the injury will occur if you get hypoxia or ischemia. It has been shown quite nicely that TNF-alpha levels in the cord blood is associated with white matter injury. High levels of TNF-alpha is seen in babies with extensive white matter injuries, and that also affects the neurodevelopmental outcome. And this TNF-alpha is released by microglia, so there's a strong connection between the two. And then if you give mice minocycline, which is an antibiotic that happens to inhibit microglia, you can reduce the hypoxic ischemic damage. So by inhibiting microglia, you can reduce the damage from asphyxia. Another way the glial cells are part of white matter injury is that hypoxia or ischemia, which leads to inflammation and cytokine release, will open up gap junctions so that the access over a large area of the brain becomes open through these gap junctions. So if you have a toxic effect in one place, those will spread to a large area quite rapidly through gap junctional signaling in the astrocytes. So astrocytes are good because they connect during development, but if you have a damage or an hypoxic insult, then they will spread the damage instead. So it works both ways, unfortunately. Another interesting thing when we talk about oligodendrocytes, the cells that myelinate the brain, if you get an hypoxic ischemic insult, you will have cells dying, like they do everywhere in the body when you get ischemia. But during development, you will have a specific pre-oligodendrocyte death if you get the insult when you have a lot of pre-oligodendrocytes during brain development. And this more severely affects the myelinization in the end. Because if you don't have any functioning precursor cells, you won't get the final cell and then you won't get the myelin. And what you can do then is to reduce the oligodendrocyte death by giving magnesium, which you can give, this is in mice, but if you give that antenatally and then the pups get ischemic damage, you will have less oligodendrocyte death, which will improve the outcome. You can also put in new astrocyte stem cells, which will save the oligodendrocytes and help with the regeneration of the damaged regions, which improves the behavioral outcome after the damage. Also studied in mice so far. You can also give the oligodendrocyte progenitor cells directly, which improves both the survival of the damaged cells, but also the survival of the animal, because you will save a lot of cells in the damaged region by putting in new ones. So the cells by themselves are really important to maintain a brain function. So my concluding remarks. I hope that I convinced you that the neuroglia are important cells in the brain, or even the most important cells in the brain, or perhaps even in the body. neuroglia are essential for neurodevelopment. Without them, the neurons doesn't function and we don't get a brain. Astrocytes regulate sleep homeostasis and the circadian rhythm, partly by light entrainment. The glymphatic system is driven by neuroglia and astrocytes and absolutely necessary for brain function. And then finally, as by the name white matter injury, neuroglia are an essential part of that as well. So I hope with this you will remember that there are neuroglia in the brain and that this is where we should focus research from now on, because we know the neurons, we've done that. Now we need to do neuroglia research instead. Thank you. Thank you.